Imagine you're staring at a tiny slice of living tissue under a microscope. What you're seeing isn't just a jumble of cells. It's a bustling metropolis, where each cell has a unique job, communicating in a language of genes. But how do scientists decode this cellular chaos? Enter single-cell RNA sequencing, a game changer that lets us study individual cells like never before. But here's the twist. While this technology gives us a treasure map of data, interpreting it is like trying to read a book in a language we only half understand. Deep learning models, powerful tools for pattern recognition, often act like black boxes, making it hard to see why they draw certain conclusions. This lack of transparency is a big deal, especially when we're talking about medicine or drug development. So how do we make these models more transparent? That's where topic modeling steps in. Think of it as organizing a library of genes into meaningful categories, like sorting books by theme. Researchers have used this to create single-cell embedded topic models, which aim to both group cells and explain their biology. But here's the problem. Existing methods often struggle with two key issues. First, they lack clear metrics to quantify how interpretable their results are. Without a standard, it's like judging a painting without knowing what good art means. Second, there's a phenomenon called interpretation collapse, where models focus too much on common genes, ignoring the rare but potentially crucial ones. It's like a chef only using salt and pepper, missing the unique spices that make a dish special. Enter scE2TM, a breakthrough framework that tackles these challenges head-on. It introduces 10 new metrics to evaluate interpretability, giving scientists a clearer lens to assess their models. But the real innovation is its approach to avoiding interpretation collapse. How? By leveraging external biological knowledge. Think of it as adding a library of expert annotations to guide the model. scE2TM also uses a technique called Embedding Clustering Regularization, ECR, which ensures topics, gene categories, stay diverse and meaningful. It's like teaching the model to look beyond the obvious, balancing common patterns with the rare, biologically significant ones. In tests across 20 datasets, scE2TM outperformed existing methods in both clustering accuracy and interpretability. It's not just faster or more precise, it's smarter about what it's learning. By bridging the gap between data and biological insight, this tool could accelerate discoveries in fields from cancer research to regenerative medicine. So the next time you hear about single-cell studies, remember, behind the data is a battle to make sense of life's smallest building blocks. And with tools like scE2TM, we're getting closer to unlocking those secrets, one cell at a time. Ever wondered how scientists unravel the secrets of individual cells? It's like trying to read a book where each page is a different cell, and the story is hidden in tiny genetic clues. Today, we're diving into a cutting-edge approach that's making sense of this cellular chaos, and it's all about interpretability. Let's start with a problem. When analyzing single-cell data, models often spit out patterns that are hard to understand. Think of it like a library with books but no titles. How do you know what each one is about? This is called interpretation collapse, where models generate overlapping, vague categories that don't really tell us anything. At its heart, scE2TM tackles a big problem. Single-cell data is too detailed. Traditional methods might tell you what genes are active, but not how they connect. Think of it like studying a city's traffic without knowing the roads. scE2TM fixes this by combining two ideas: cell embeddings, a mathematical map of cells, and topic modeling, grouping genes into themes like immune response or energy production. It's like having a GPS that not only shows you the city, but also explains why certain streets are busy. Here's where it gets smart. The model uses a multinomial distribution. Imagine rolling a dice where each face represents a gene's activity level. This helps it guess how genes might behave in different cell states. But it doesn't stop there. A loss function balances two goals: reconstructing cells accurately like following a map, and keeping the model interpretable, avoiding traffic jams by sticking to main roads. This prevents a common pitfall: interpretation collapse, where models become so complex they lose their biological meaning. Think of the loss function as a GPS. One part guides the model to rebuild cells correctly, while the other ensures it doesn't get lost in complexity. By combining these, scE2TM avoids the black box problem. Other models might give answers, but scE2TM explains why. Now, why does this matter? Earlier methods like scVI or scDCC focused on gene expression patterns, but ignored the network of interactions between cells, like studying a city's traffic without considering its roads. scE2TM fixes this by integrating external knowledge from a foundation model, a pre-trained AI that knows biology. This lets it learn from data and existing scientific insights, making it smarter and more reliable. What's truly groundbreaking is how scE2TM handles interpretability. Unlike black box models, it uses topic modeling to label gene activity with biological meanings. For example, it might identify a group of genes linked to cell division or stress responses, then map those patterns back to real-world functions. This isn't just data, it's a window into how cells work. And the results? They're huge. In tests, scE2TM outperformed older methods in clustering accuracy, grouping similar cells with precision. But here's the kicker: it also uncovered biologically meaningful insights. In a human pancreas dataset, it revealed topics tied to insulin production and cell communication, findings that matched experimental data. It's like having a translator for the genetic code, turning raw numbers into stories about health and disease. So what's next? This tool could revolutionize how we study diseases, identify novel cell types, and even design targeted therapies. The future of single-cell biology isn't just about data, it's about understanding it. And with scE2TM, we're one step closer to decoding life's most intricate code.